Good afternoon, and thank you for joining us for this Chemistry World webinar. It's delightful to have you with us. These sorts of events we find are at their best when they are interactive. So not only will you see and hear the presentation going on, but we want you to get involved in the polls and we want you to ask questions as well. So before I tell you more about what we're actually going to be covering today, just want to make sure that you understand how you can use GoToWebinar, the platform, to ask questions as and when they occur to you. Now, GoToWebinar loads up differently for different people, depending on what device you're on, but generally it'll be a strip down one side of your screen, and at the bottom there will be a chat box in which you can put some questions. Now, I've already sent a message out in there that you may have seen when you came in, so that's where you get your questions in, and your questions are an important part of this. We have two excellent speakers with us who are more than happy to answer your questions after the presentation, so do please get them in as soon as they occur to you. Now, as a little thank you to everybody who has attended live today, we will be sending you a certificate of attendance, which will land in your inbox over the course of the next couple of days. And in the same email, there will be a link to a recorded version of this webinar. Now, often people ask us if we share the slides from the presentation. We think that it's more important that you get the slides in the context of what's being said. And so rather than just sharing a slide deck, we'll send you a copy of the recording, and that way if there's anything you missed, anything you'd like to go over again, any detail or reference you'd like to look up, you can just find the relevant bit in the recording and get the important context of what's being said as well as what appeared on the screen at the time. So keep an eye out in your inbox for a link to that recording as well, in the same email as your certificate. That's the other thing people ask us about all the time, so keep an eye out for that and do keep your questions coming in. So I'm Ben Valsler, Chemistry World's digital editor. My role today is just to introduce our guests, introduce the topic, and then be your voice for the questions coming up later. So today we're going to learn about synthesis and atomic level characterization of advanced zeolite-based catalysts. And we've partnered up with Thermo Fisher Scientific in order to bring you the right people to talk about this topic. I've no doubt that Thermo Fisher will be a familiar name to you anyway, but their mission is to enable their customers to make the world healthier, cleaner, and safer. And they do that by helping people to accelerate material science research, solve complex analytical challenges, and to increase productivity. And it's those complex analytical challenges that we're looking at today, because we'll get an introduction to the relevance of materials in catalysis, the challenges in their synthesis, and their characterization at atomic level. We'll see a protocol to determine the location of different types of metallic species within a zeolite framework and the quantification of the metal-metal interactions. We'll explore experimental issues, analysis of the experimental images using AI techniques that are based on clustering, and we'll use the help of special fault holders, which along with image simulations will help you gain a better understanding. Now, our two speakers, you can see their pictures and names on screen right now, but they are Jose Calvino, who's a professor of inorganic chemistry, was formerly president of the Spanish Society of Microscopy, and since 2004 he's been leading the electron microscopy facilities at the University of Cadiz, Spain, where also his colleague Miguel works. Now, Miguel Lopez Haro is a assistant professor for inorganic chemistry. He's now responsible for a new research line at the university focused on quantitative methodologies for the structural and compositional analysis of functional materials. So they are exactly the right people to handle this sort of topic for you. Now, we're going to get them up on camera and on screen in just a second, but before that, one final reminder of how you ask questions. Down at the bottom of the GoToWebinar panel, get your questions in, and then after the presentations, we're also going to have a couple of polls that help us to get to know you as an audience and to understand how we can best follow up with you and best supply you with the content that you need in this Chemistry World webinar series. So let's get Jose and Miguel on camera. I think we'll be starting with Jose. So let's get them up. Hopefully we'll see their screen and we'll see their webcam very shortly. And then obviously we'll have to check that we can hear them. We can certainly hear something, so that's a very good start.
Okay.
We can see their screen, that's great. Can't see your webcam yet, Jose. And there we go.
Good afternoon, everybody.
Thank you for joining us.
Thank you. Thank you for this kind introduction to the subject and also to the two guys. We are here to talk about this topic, okay? Thanks a lot.
Excellent. Well, I shall leave you to it. Take it away and keep an eye out for those questions. Jose and Miguel, as I said, very kindly have offered to answer any and all of your questions at the end of the presentation, but that doesn't mean you should wait until then. Type in your questions as soon as they occur to you, and it gives us the maximum chance of getting through as many questions as possible. So enjoy the presentation. I'll be back with Q&A after Jose and Miguel. Thank you again.
So good afternoon, everybody. So first of all, I would like to thank Thermo Fisher for thinking about this webinar, and for, of course, providing support to prepare it, and also Royal Society of Chemistry for hosting it. So today, in this afternoon, we will be talking, as Ben said, about the use of STEM techniques to characterize the structure of bimetallic catalysts embedded in zeolitic matrices, and we will make emphasis on the quantitative part of this work. And by quantitative, we want to mean that we want to obtain data which are representative of the material as a whole, not just having a look at the very fine details at atomic scale, but having numbers and figures out of our characterization study, which can be representing the behavior of the material as a whole. And I think that in the case of catalysis, this is in fact one of the big challenges of microscopy in general, not the microscopy of bimetallic catalysts in zeolite, but in general of microscopy of catalysts. Okay, so but before going further, I want to make clear that this is a collaboration work. The materials and the catalytic concepts have been put forward by the group of Professor Avelino Corma in the Institute of Chemical Technology in Valencia, and they have particularly Dr. Lishan Liu has been enrolled very closely to us in the development of all the methodologies that we will be talking about this afternoon. Thinking about quantitative things, for sure, very important to compare microscopy with microscopic data, and in this case, all the materials we have been investigating have also been followed by XAFs and XANEs using the installation of the alpha synchrotron radiation light source in Barcelona. Okay, and I would also like to thank one of our colleagues here in Cadiz, Dr. Ramon Mansorro, who has participated in the part of the work related to quasi-in situ experiments. So the outline of our presentation today will be more or less this, the one I'm showing here. We will first start talking a little bit about the materials, what are the challenges from the catalytic and the synthesis points of view. Then we will move on to talk a little bit about which are the particular challenges of STEM characterization of this very specific type of materials, and characterization, as I said before, in a way that we can get some statistical significance from the data we are acquiring in the microscope. This is quite important for us, and we also want to have data which are really reliable in the sense that they represent the actual structure of the material. Then, well, we'll think this will be my topic here. So then we will move on to the part that will be presented by my colleague, Dr. Lopez Aro. He will give you the details that you want to hear about the methodology to characterize in quantitative terms and atomic level subnanometric bimetallic nanoclusters dispersed in the matrix of a particular type of zeolite, the MFI zeolite. And you will see here how important it is to use simultaneously HAADF and iDPC imaging in combination with image simulation, and for sure trying to approach the analysis of all the data using machine learning techniques, particularly using clustering techniques like that based on the so-called k-means. Okay, We will be talking about materials we have been prepared by reduction in hydrogen. But after that, and before going into the microscope in conventional experiments, we have to go through the air and put the samples in contact with CO2, oxygen, and water. And this can make some small modifications of the actual structure of the catalyst. So we will pay also some attention about the influence of sample air interactions in our observations. And in this sense, we will end up our presentation talking a little bit about anaerobic transfer experiments and how these anaerobic transfer experiments correlate with those performed under, let's say, conventional ex situ conditions. And finally, we will end up with some conclusions. So starting with the materials, we should recall that platinum itself is a metal which is able to activate very nicely hydrogen and carbon-hydrogen bonds, and it does not activate so well carbon-carbon bonds. So in principle, it's a good candidate as a metal for a catalyst if we want to use this catalyst in reactions related to selective hydrogenation or dehydrogenation reactions. And well, in the papers that you will read about this presentation today, the focus of the catalyst we are going to talk about is particularly on dehydrogenation of the use of these materials as catalysts in dehydrogenation of propene to propylene. Okay, so but well, we will be talking here in general about platinum-based catalysts. From the point of view of catalysis, for sure, the challenges are always to have a very active catalyst. In this case, a very selective catalyst, in the sense that it breaks particularly carbon-hydrogen bonds, and it does not make any carbon-carbon breaking work. So we want it to be active, selective, and for sure we want our catalyst to be to long forever and to keep active forever. So we want it to be stable. Okay, and to achieve these functionalities challenges, it's very important to control different parameters of the catalyst. For example, it's very important to control the dispersion of the metallic phase, and in this sense, it is well known that for this kind of reactions, the species that perform the best are in general subnanometric size platinum clusters with diameters in the range of, let's say, half a nanometer or slightly below the nanometer. To control both activity and selectivity, one of the major challenges is to control, in fact, the electronic state of the platinum atoms in our catalyst. And this for sure will depend on both on, for sure, dispersion, but also very importantly on the interactions with the metal establishes with another metal, which we can add on purpose to control electronic state, and for sure with the support of the metallic particles. Okay, and finally, concerning the activation or stability issues, the activation takes place mostly through sintering. This is particle size, particle agglomeration and increase of particle size. Or in this case, when there is a lot of carbon-carbon breakage, we can have, and carbon H breakage, we can have the formation of coke, and this coke will avoid the contact with the reactants, and this will make our catalyst to reactivate. And to avoid this these problems, we should take care of migration of platinum in the catalyst, and for sure we should also take care of the acidity of the upper surface, just to minimize this carbon-carbon breaking. So zeolites provide the environment necessary to take control, or at least have a synthetic idea which allow us to take control on all these questions that I have posed here. So we in a zeolitic matrix, we have pores with size in the range of a few nanometers, we have different kind of pores. So in principle, we could try to accommodate the metallic entities inside these pores, and maybe these pores can act as can avoid migration out of the pore and therefore control the influence of platinum migration and therefore sintering. Okay, we can also control by, well, this is what we can call confinement effects for sure. We can add some additive to the zeolite, for example, potassium ions, and with these potassium ions we can modify somehow, as we will see, the ability of platinum both to migrate and to sinter. And also, for sure, we will be able to control the acidity of the zeolite. So adding this modifier is an important issue in the synthesis. And for sure, if we want to have a very well-defined electronic state for platinum, we should deposit or locate these metallic entities in specific sites where the environment is very well controlled, and where we could add, for example, a second metal. In our case, we will be talking about tin, and we can control in that way by putting it in certain locations, this kind of interactions. Okay, so from a synthetic point of view, the challenge would be to disperse bimetallic clusters in the subnanometric range size regioselectively in particular locations of the zeolite, and this can be done by controlling in a one-pot synthesis the template that we are using to create the framework structure. We can add potassium to control the behavior of the metallic entities during the activation procedures. As you can see here, we don't use potassium. We can get sintering problems during calcination or during the reduction, which is usually applied in all these catalysts before using them in the catalytic reaction. So from the point of view of synthesis, this is the challenge: putting subnanometric cluster in a specific sites in contact with a second metal and in the presence also of potassium. So we want to do this. The people in the ITQ, they have managed to do this in a very nice way. But the question that comes now is, once we have designed our synthesis, once we have performed our synthesis, is everything as we expected? And this is the point where microscopy comes in, and this is the point where microscopists are asked to say, okay, did we get the goal that we posed on the synthesis or not? Well, and this is the challenge for us as microscopists. So we have to answer different questions. We have to detect the subnanometer cluster. We have to determine their location in the porous structure, and we have to determine if the two metals are in contact or not, or if they are separated. So these are the different questions that the microscopists have to answer, and we have to answer with a very special type of material. We have a matrix, zeolitic materials in general, which are very unstable under the electron beam, so we don't have a large chance to be observing the material for long with long exposure time. So this is forbidden if you want to get something which is just pristine material. And we have to determine what the bimetallic clusters are in a porous structure which is really complex, which is presenting different type of pores, and we have to determine where the bimetallic entities are located. So these are the two major challenges that we have to face. There are some others for sure that you will see in a few minutes, but these are the two major ones, and the ones that we have tried to approach in this paper that is just shown on the bottom part of the slide. So our approach has been to benefit from simultaneous STEM imaging using HAADF and iDPC. Okay, most of you know that the high angle annular dark field imaging is a technique which is sensitive to the atomic number of the elements which are present in the material, and this the contrast in these images are roughly related to the square of the atomic number. So if you have a look here about the elements, the set numbers of the elements we have in our case, we have platinum 79, tin 50, and very light elements. So this technique will be very sensitive to imaging the metallic part of the catalyst. But in general, if we want to work at conditions where we are not damaging the material, we have to work under conditions where we will have a very low signal to noise ratio, and this makes visualizing the structure of the zeolite matrix very complicated. We cannot say very well where the metallic entities are just on the base of observing the HAADF images. So we take advantage of the set proportionality in the contrast of this iDPC imaging technique, and moreover, we will take benefit from the high signal to noise ratio characteristic of this technique, which will allow us to work under very low dose conditions, which are able to detect the metal on the HAADF detector, and also to have a very nice look at the pore structure of the zeolite. So by correlating these pairs of images, and this is why we call it HAADF iDPC pairs, we will be able, as Miguel will show you, to learn about the questions we want to clarify. So this looks very simple, but I mean the whole process, it takes a lot of preparation. It takes a lot of optimization. We have to start optimizing from the very beginning. Very simple questions like sample preparation, the choice of solvent to disperse our sample, the type of holy carbon we're going to use. We have to, for sure, optimize the conditions where we are going to record our images, and this is a trial and error. Work where we have to spend quite a lot of time just looking for the best conditions to make possible this work. And finally, a very important part is what to do and how to process all the information that we will be obtaining in the microscope in a way that we can make an statistical analysis. And this makes a lot of work related to the noise in our images and preparing our images to measure, and then using techniques to quantify, to get numbers in an automated way without any bias from the microscopy, I would say, so that everything is done automatically and we can get a lot of data to treat all the information in a reliable and in a statistical way. Well, this is just a short presentation about the protocol that we have prepared. So now I hand over the presentation to Miguel, and he's the one who really knows the subject, and who will tell you all the details about this protocol. Okay, so he will concentrate on these last two parts of the methodology. So if anyone has questions regarding the first one, we will be very happy to answer. So Miguel.
So thank you very much, Jose, for your very nice introduction. So now I will continue with the webinar, and of course I will focus in these two parts. So here what I'm showing is just HAADF iDPC per image recorded in a medium or high resolution. So this is HAADF in blue. This is on the right part is the iDPC, and it's recording, and I will show how it affects the dose what we are using when we are working with this kind of material. So this is a fresh picture, and then if we take a second picture, you will see that in this area, for example, if we focus in this area, we can start to see some degradation. Of course, if the zeolite structure become degraded, the position of the cluster will move again. So if we take a second or a third picture of the area, this area become more damages than before, and the fourth one you will see that is already damaged. So what does it mean? That means that the zeolite is really sensitive to the electron beam. So in order to get a very good picture, we have just one opportunity, only we have one shot, and for that we have to tune very well the electron dose. So the question is, how do we can control this electron dose in the microscope? So in order to control that, we have to tune first the beam current, then the dual time. The time is so we have an electron probe that is scanning our sample, and the dual time is the time that this probe is staying in 1 pixel. So faster is this time, so lower is this time. Less time will be the probe in the zeolite, and of course the damage will be less important. The third point that is important to control the dose of our experiment is the magnification at the image size. And the fourth, the last point, but it's not related to the electron dose itself, but it's also important related to the visualization of the cluster and single species, is the depth of focus that is rated mostly by the conversion angle of our microscope. Okay, so once that we can control our dose, we can record our images without any problem of damaging. But remember, we are using HAADF detector and DF4 or iDPC detector. So as we are working at very low dose, even using HAADF is still noisy. So in order to improve the visibility of our cluster, we have to apply techniques that allow to remove the noise. So this technique in this work that we have been using is a technique based on undecimated Waring transform, combining unconvalescent stabilization. So here we have our raw HAADF images recording at the same time the iDPC, of course. So after applying our filter to the noise, the image, we can see very clearly the species, the metal species located in our frame of the zeolite, of course. So look at that. That, for example, here we have a high contrast that can be also detected in the raw image, but it's more difficult to see. And then, in order to boost the intensity of our cluster, what we are doing is to remove the background of the image. So this is the final treatment that we are using in order to improve the visibility of the cluster. So first, in order to get a good picture, we have to tune our dose, electron dose, and the second one, we are using image processing to remove the noise coming from the HAADF detector. So by combining these two things, we are able to clearly see the single or the subnanometric cluster. Okay, so once that we have our condition ready, we need to understand the crystallographic structure of our material. So here I show the model extracted from the zeolite database, ETHA, of a specific zeolite, that's MFI. This is the zeolite that we are studying in this webinar. So this structure is along the O1 orientation, okay? And we can distinguish 3 different parts, okay? The straight membrane that has this size. We can distinguish the intersectional void that are located in this circle, okay? By marking this circle, that having that size, okay, around 5 nanometer in diameter. And also we can distinguish sinusoidal membrane that are located just where this line are marked. This orientation is the most typical one that we can find in our experiment due to the way that we are preparing our sample on top of the nickel carbon grid or copper carbon grid, okay? However, we can find also another orientation, the 100 and the 001, but this is not so common to see. And also I would like to point out the importance of show when the zeolite are off axis. Okay, that means we have the crystals sometimes are really well lie on the carbon layer, but sometimes this is a little bit tilted. When they are tilted, we are not having the pore well orientated, and we are finding our HAADF iDPC pair images this kind of line. So taking account this structure, we go to STEM iDPC, and here we show one experimental image recording by this low dose condition in along 010 zone axis. Okay, and if we took a zoom from one part of the image, know that we are able to distinguish the different part that the model are telling us. Okay, so we can distinguish very well the straighter membrane that are located here, this black hole. We are able to distinguish the intersectional voids that are clearly here in this orange circle, and also we are able to distinguish the sinusoidal membrane. Okay, so as we are able to distinguish the different channels that this MF5 has, we are able to then by combining HAADF, we are able to localize directly the position of our clusters. So once that our image conditions are ready, our denoise is done, and we know the structure of the zeolite, we can directly interpret, or we can directly identify the location of the cluster. Here I show experimental images of a highly dispersed platinum species on top of, inside of the MF5 zeolite. Okay, so this is the HAADF, the treated HAADF image, and this is the iDPC. So by correlating the location of these high contrast species, we are in the iDPC, we are able to distinguish where are located inside of the zeolite. And here I show similar images, treated HAADF images and iDPC images but for subnanometric clusters. If we take a zoom, for example, in this area, we can clearly see where the clusters are located inside of this framework. Okay, look at that always we are showing images in zone axis and images in out of axis. Okay, because I will show you before, help us a lot to localize the cluster inside of the zeolite. So once that we're able to localize this cluster that are normally located in the sinusoidal channel, okay, according to the structure that we have already described. The second question is, okay, I know where the clusters are, but I would like to know the chemical or the elemental identification of these species. Okay, so looking at the HAADF images at high resolution, we're able to distinguish the experimental images after treat. Then two kind of contrast. Contrast coming from possible platinum because it's very bright one, and lowest contrast, so lower than the platinum, that can be tin. If we remember, the atomic number from platinum is 79 and the atomic number from tin is 50. Compared to silicon and oxygen, in HAADF can be possible to distinguish them. However, we have to remember that intensity in HAADF images is not just proportional to atomic number, but is also mass thickness dependent. What does it mean that? That means that if our zeolite is not completely flat, the surface, that can give us thickness increment in some local areas. And in the HAADF, this increment of the thickness will appear at high contrast. So in order to be able to distinguish this high contrast from the platinum or tin, or if this contrast is coming from some artifact from the zeolite itself, we need to carry out some other techniques. In addition, if we also take a zoom of this cluster, we can see also that in some areas the platinum has some asymmetry or some tail, where the intensity is much lower than the cluster that can belong to the platinum. Okay, we can see if we take, for example, an intensity profile along this rectangle. So this is the atomic profile. So also we can expect that this is thin in contact with platinum, but we are not sure because some artifacts coming from the HAADF image. So the first technique that one can think about is EDX. Of course, EDX, or X-ray energy dispersion spectroscopy, can be a very useful technique to identify the chemical nature of our species. But when we are working at very high or high magnification, there is a big problem. First, when this element has a very weak characteristic signal, even if we are using or we are working with single atom. So the EDX detector right now, even using this nice four quadrant EDX detector, are not able to distinguish very clear the signal from one single atom. Also, the signal is corrupted by noise. And the third one is the influence of the atomic displacement induced by the thermal excitation and the electron beam. That means that we are recording our images, and the probe is scanning our sample. We can find some movement due to this thermal excitation. And the fourth one that is not written in this list is, remember, we are working with zeolite. They are really being sensitive material. So in EDX, normally you have to increase a lot the beam current to get a nice signal. So if we are working at high resolution and we increase the beam current, we are increasing the dose, and at the end, what we are doing is destroying our sample. So in order to be able to understand that the contrast that we have in our HAADF image, what we have done is, okay, let's do some simulation. For that, we create models first in the zone axis, in the main zone axis that we can detect in our experimental HAADF iDPC per images. So here we have the model of the zeolite in 101. And inside of this model, we have introduced some platinum cluster having different size, one single atoms, some thin cluster having also different size, and one single thin atom. Then we have used a specific software to carry out the simulation. We have used this optoelectronic parameter close to those, or very close to the parameter that we are using experimentally, and we obtain our high resolution image simulation. Okay, but remember, we are working a very low dose, so our high HAADF image is really noisy. So in order to understand the effect of the noise in our simulation, we have added two kinds of noise: Poisson noise and Gaussian white noise. Okay, these two kinds of noise are the most important in the HAADF image that we are recording experimentally. So once that we have introduced the noise, we have applied our filter to remove it and to clean up the image. And of course, we have removed the background in order to apply the same protocol that we are using for our experimental image. So something that is very important I would like to highlight is that after apply the noise technique, we're able to recover the same intensity that we have before adding noise. Okay, so for example, this cluster where they belong to thin, once that introduce the noise, the intensity decrease a bit. But once that we are denoising, know that we are obtaining the same level of intensity. That is very important in order that allow us, but that by using this denoising technique, we're able to show up everything that we have in our, or almost everything that we have in our zeolite. But now that the thin cluster are clearly visible after the simulation, but when we are in the zone axis, know that some thin cluster are also possible to distinguish them, but the the platinum atom is almost disappear, and the contrast that appear after even denoising and removing the background is close to those contrasts that the zeolite has in the framework. And remember that also we have put tin single atom that is completely vanished in the simulation. So by using these HAADF images, we can conclude that, okay, we can distinguish tin cluster and platinum cluster. Platinum single atom is hard to distinguish from the zeolite, but it's still some contrast appear, but we cannot distinguish tin single atom in this orientation, okay? So if you remember, we are also working with off-axis HAADF images, and of course we have simulated. What we have done is just tilt this model 15 degrees, and we are our image in that, like that, okay? We don't have a channel well oriented anymore, and we are also putting the same cluster in the same position. We are using the same of the electronic parameter, and we have carried out the same protocol, okay, of adding noise, the image, and background subtracting the noise image, okay. And what we can see here is that the single and the cluster from tin and from platinum are clearly detected. Of course, this is because we are avoiding what is called channeling effect. So as we are avoiding this effect, we can improve the signal of these tiny species that are inside of the zeolite. But it's still tin single atom is not detectable. Okay, so from this simulation we can see that we're able to distinguish the nature of our cluster, but the limitation is that we are not able to distinguish or to detect single tin single atoms. That is a very important conclusion for the next slide. Okay, so once that we know that we can distinguish the species, the next question is, but we can distinguish if there is some contact between platinum and tin. So for that also we did HAADF image simulation, but for that, of course, as we already know that the off-axis images are better than the on-axis one, we have carried out the simulation in off-axis images. So for that, first, in a very small area, in order to speed up the simulation, we have just put it one single cluster of platinum. In this model, what we have put is one cluster of platinum in contact. I don't know if you can see clear or not, or is it too small, the figure, in contact with one single tin atom. In the third model, what we have created is a pure contact of two clusters. One belongs to platinum, another one belongs to tin. Okay, similar model, but increasing the size of the cluster has been done, and we have carried out the simulation using this of the electronic parameter that we already know from our microscope. So if we perform line profile along this image simulation, okay, we can see that when some clusters are in contact, some asymmetry from the profile appear. Okay, so this is very important because if you remember in the previous slide, I showed that some asymmetry in the intensity profile appear also. So thanks to this simulation, we can see that platinum and tin can be in contact. So by using this simulation, first we're able to distinguish the nature of our cluster, and also we're able to detect platinum tin contact at atomic resolution. However, this is just analysis of a few clusters, and what we want to get is a statistical information of this contact. Okay, so in order to carry out that, what we have done is segmentate the HAADF images by using k-mean clustering method. But in order to know if this method is working or not in the experimental images, before doing that, we decide to perform this analysis on the image simulation. Why? Because we already know where the clusters are and the nature of this cluster. So by using this k-mean clustering, that is a unsupervised matching algorithm, unsupervised machine learning algorithm, we have able to segment the intensities of the HAADF image simulation in zone axis and in off axis, okay, tilted one. So we have applied the k-mean segmentation, and we found that the best number of k-mean cluster is 10. That means that we can group. This means that we can group the intensity of our whole image in 10 representative clusters. And once that we are able to separate the intensity in 10 clusters, then we can say, okay, give me the cluster that belong to tin and give me the cluster that belong to platinum. Also, we have studied the effect of how the noise is affected to this analysis. Because of that, we have four k-mean maps, okay? Because we have been studying the effect of the tin, sorry, of the noise, and for images in axis, in off axis, sorry, and images in off axis. And now that we're able to separate very clearly those, at least those species that belong to cluster from tin and from platinum. In some images, we are also able to distinguish or appear the signal that belong to the platinum. But as you can see, this is misleading, because this signal that we know that belong to the single atom of platinum that is located here, the map says that this belongs to the tin. So, but of course, we are not able that it was located here, the tin, we are not able to distinguish. Okay, so if we do the same for the off-axis of a tilt images, we are also to see more clearly the species that we have in our samples. But note that here what we are showing that sometimes the platinum single atom is identified as tin cluster. It's not a perfect method, but it can help us to get a statistical view of our analysis. So in order to perform this k-mean cluster analysis, we perform an experiment in quasi-in situ mode. Why? Because we know that when these platinum tin M5 sample are exposed, are in contact with air, the tin is oxidized, and we wanted to see exactly the real platinum tin contact. So in order to avoid the oxygen, what we did, we designed an experiment where we performing reduction at different time inside of a reactor. The reactor can be closed by using this bubble, okay? And we can transfer this reactor to the glove box. Inside of the glove box, we have a specific holder that allow us to transfer the sample inside of this chamber to our microscope. So if everything goes well, that is the sample is totally is not having any contact with the air. So we preserve the nature of the platinum and the tint as we can expect in the reaction. Okay. So okay, we did this treatment for different hours, and we perform the HAADF iDPC per images. We clean up by using this denoise technique, and we apply k-mean segmentation for sample that has not been in contact with air. and with sample that has been in contact with air. Those samples that has not been, that the samples that have not been in contact with air, we have been called like quasi in situ, okay, and in contact with air, we call ex situ. Now that after using this coming cluster number, we're able to distinguish orange areas that belong to platinum and green areas that belong to tin. And know that the location of these species are completely different when the sample is in contact with air and when the sample is not in contact with air. Look at that. This is the same image, okay, but just in a zoom to better show that features. Okay, so in order to so once that we have segmented that, we can quantify the contact of tin and platinum system. How? First, I have to show that in order to have an idea of the contact, we carry out an excess experiment, and we saw that the platinum and the platinum and the tin are totally in contact. However, if we want to measure the distance between tin and platinum in our coming images, what we are measuring is not from the surface of the particle. We are measuring from the center of our feature that we have in our image. So for that, we decide to use a threshold of 0.2 nanometer of distance. So taking this threshold into account, we perform an histogram of number of contact. So in that case, we analyze more than 10 high-resolution SDF STEM images. Normally, all of them has been analyzed in off-axis in order to avoid any problem with the challenge effect. And for sample treated at 0 hour and sample treated to 22 hours. Okay, for ex situ catalyst and for quasi in situ catalyst. What we can see from this histogram is that the number of contact for quasi in situ experiment increase when the reduction treatment increase in the time. So also what we can do is, okay, this is our histogram, but we can calculate the percentage of number of platinum clusters that are in contact with the tin species. So if we represent or if we plot this platinum contact, we can see very clearly how when the samples are treated ex situ, there is an increase for after there is the contact increase after 3 hours of treatment, but then this there is a decreasing in number of contacts. However, this platinum contact can increase when the experiment has been carried out in quasi in situ. This is a very important data that has been compared not just by other microscopy data, but also we have compared the result with a CO chemistry experiment, and these two data match very well and allow us to understand how is the nature of this catalyst at atomic scale. So just to finish our webinar, that we are on time, I would like to show the conclusion. So first, the use of the complementary STEM-based technique has allowed us to identify the location of synonymous metal species encapsulated in a pure siliceous M5 structure. Okay, the chemical nature of the platinum and tin synonymic cluster and the distribution within this specific zeolite has been estimated by using the means clustering algorithm, and of course, combining the STEM images with machine learning algorithm, in particular this coming and image model simulation, we have been able to correlate or to understand the structure and the reactivity for this important system that is used in the industrial for selective dehydrogenation of propylene to propylene reaction. In any case, thank you very much for your attention, and for more information here I would like to show the different reference with having more and more specific information. So thank you very much. And now we are open for questions.
Thank you very much to both of you. What we'll do, it's tricky to copy references down from a slide. What we'll do is we will copy these references ourselves and we'll put them on the webpage at chemistryworld.com/webinar. So the page that you actually initially registered from, what we'll do is in fact send you a link now in GoToWebinar, and we'll paste all of those references in there for you to find, because that saves you a lot of quick copying and typing. So the link for the Chemistry World page has just been shared in the GoToWebinar chat now, and we'll put all of those references in there for you very shortly after the webinar. Now, before we go into a Q&A, we're going to launch a couple of polls. We're going to find out a little bit more about you as an audience. So let's get the first one up now, and we will just give you 30 seconds or so to answer. So what is your biggest characterization challenge? Is it imaging quality, so resolution and contrast, and that's where things like the noise reduction we've seen today would come into play? Is it sample integrity? Do you find your samples are just too sensitive to air or too easily damaged by the microscope beam itself? Is it data processing and throughput? Is it preparing your sample in the first place, or is it something else? And you can tick more than one. So do please let us know. This helps us to shape future webinars with partners like Thermo Fisher, which will allow us to make sure that we're giving you the information that you need and they're supplying the answers that you need as well. We will still have a couple of minutes of questions at the end, so do keep your questions coming in. We have had some excellent questions already. May not get time to get through them all, but we'll do our best and get through as many as we can. And to those of you who are just saying thank you in the chat, well, thank you for joining us. Let's give you a few more seconds to vote in this so we really get a good idea of what your characterization challenges currently are, and then we will close that down and move on. So I'm going to make sure at least half of you have had a chance to vote, and we're on 46% now. So if you haven't voted in the poll yet, this is your last few seconds to make sure that you get your voice heard in that poll. And there we go, we've hit 50%. So I'm going to close that, and then just purely out of your own interest, you can see now most people, it's sample integrity, imaging quality, and data processing that are the challenges. Sample preparation and other issues less so. So that really helps us in future to make sure that we're giving you the information that you need. Thank you for that. And one more very quick poll just to help us know who it is that we should get in touch with. Let's launch the next poll now, and I will just start to go through some questions while you answer this one. So simply, would you like a tech specialist from Thermo to get in touch with you to help you resolve some of those issues that you may have just put forward there? Now, we will dive into some questions in the last few minutes that we have. There were quite a lot of questions that came through that I think are ideally for you, Miguel, that were to do with the imaging, image processing, and so on. So let's start with a really simple technical one from Donald, who asked, what software has been used for the image processing?
So what we are using, Velox for recording the image on the microscope, and then we have made our homemade script by MATLAB.
Excellent. There we go, Donald. Thank you for your question. Sticking with the images, Shantanu asked, are the images probe corrected, and what probe corrector is the microscope equipped with?
Sorry, say again?
The images, are they probe corrected?
Yeah, of course, they are probe corrected one. So we have just working with a probe corrector. Our microscope actually is a double corrected microscope. So yeah, you need to use this probe corrector microscope to get a very nice resolution.
Excellent. And I think this will probably be the last one specifically about the images. Let's dig out. Actually, no, let's move on to techniques. So this is one from Sub, who says, through these techniques, can we capture 3D images of these catalysts, or are they only 2D planar structures that we can get?
This is a very good question. So it's actually that we are working now. So It is possible, but it's really hard just to show if the clusters are inside or not by using a very, very fast tomography experiment. And also, if the iDPC image is monotonic with the intensity, it's also possible to use it. But it's something that we are working now. Actually, we have a project working on that, so we can answer that in a couple of years.
Well, good luck with the project, and hopefully we'll be able to come back for another webinar and you can show us all about how that works as well. So Jose, let's talk about the technique more broadly. Obviously, this was a specific example dealing with specific problems. How would we extend it to other metal zeolite materials?
Okay, this is also a nice question. I mean, in principle, the approach we have put forward, I think it should be, I mean, this HAADF iDPC pair images using these clustering techniques and these simulations, this should be in principle of application to any kind of metal embedded in a zeolitic matrix. For sure, every problem has to be taken into account on its own, so it's very important to make simulations of the specific problem. It will depend on the type of pores that the zeolite may have. What are the most common projections in which this material will be imaged, and what are the possibilities to distinguish metal and matrix in those projections. But in general, it's a bit of a large application to many other type of let's say metals or bimetallic systems in I would say in being sensitive materials. So the improvement in detector is being so fast now, and I mean, I would say that right now our microscope is an old one in some sense. So taking into account the improvement in the last few years. So I think that there are great expectations to do this kind of work on even much more sensitive materials than the one we have tried. For example, we have tried a pure siliceous matrix, and this is being sensitive, but not so sensitive as those containing aluminum. With the new detectors, maybe you can also explore aluminum silicon matrices, MOFs and COFs, and this kind of materials which are even more sensitive than the one we have studied. So I think that the opportunities are pretty large.
Excellent, thank you. Well, we've technically run over time. Thank you to everybody who has stuck with us, and to everybody who's asked a question that we didn't get through. I'm sorry, so many good questions. Thank you. We really appreciate it all. So we do have a record of them that I will send over to Thermo Fisher, so they may be able to get in touch and help answer some of your questions anyway. Speaking of which, some of you may have had questions answered in text by our partner from Thermo, Min Wu, who's been in the background helping us to go through all of these questions. Thank you, Min, for joining us as well. But obviously, importantly, thank you to Jose Calvino and Miguel Lopez Haro from the University of Cadiz, who have joined us for today's presentation. Thank you to Thermo for arranging this for us, and thank you to you, the audience. One final question that has come through that I think I answered at the beginning, which was from Sabura, who asked, can I get a certificate of attendance? Well, yes, everybody who has attended the live webinar will get a certificate in their inbox in the course of the next couple of days, along with a link to the recording. So if there's anything that Jose or Miguel said that you'd like to go back and watch again, then you can see that in the recording that will be in your inbox in a matter of a couple of days' time. So that's it for this Chemistry World webinar. Thank you once again to Jose and Miguel. Thank you to Thermo Fisher for putting us in touch with them and for highlighting this as an important topic for us to talk about. I'm Ben Valsler, Chemistry World's digital editor, and I will be back again for another Chemistry World webinar very soon. Have a look at chemistryworld.com/webinars to find out what we've got coming up. Thanks again, and see you again soon.